In this video, I want to talk about the similarities and differences between the electron transport chain and the photosynthetic light reaction specifically. So they have some similarities, some things in common. Both of them have electron flow, and this electron flow is through a chain of membrane-bound carriers that includes the quinones, cytochromes, iron-sulfur clusters, etc. So the fact that there's electron flow means both of these processes have redox reactions. Now, these redox reactions, many of them typically are spontaneous and exergonic, right? And photosynthesis and the electron transport chain differ slightly in this regard, but that's something I'll get to when I talk about the differences. But both of them have electron flow through a chain of membrane-bound carriers, and they both have redox reactions. In addition, both of them use these redox reactions to establish proton gradients, which are also pH gradients. And these gradients are across a membrane. So what happens here is the downhill flow of electrons, right? The spontaneous flow of electrons through the redox reactions drives the uphill flow of protons, right? The protons are being pumped into a region that has a high concentration of protons. So these protons are moving against their gradient, and these electrons are moving downhill in that they're moving to higher reduction potentials. So both establish proton gradients across a membrane through the spontaneous flow of electrons. Once both of them have these established proton gradients, they both couple the downhill flow of protons back across the membrane through specific protein channels. In the mitochondria, that protein channel is the F0 subunit, and in chloroplasts it's the CF0 subunit. And then with phosphorylation of ADP to make ATP, which is done by the F1 subunit in mitochondria and the CF1 subunit in chloroplasts. In both cases, we're talking about ATP synthase, right? Those subunits are part of the ATP synthase enzyme, which actually makes the ATPs. So they both have this chemiosmotic coupling. They both use the spontaneous flow of protons back down their gradients to power the production of ATP. Some differences about them, though. So what are the initial and terminal electron donors and carriers, respectively, in both cases? In mitochondria, we have initial donors. The initial electron donors are reduced coenzymes, and if you recall, those are NADH and FADH2. Now, these two are willing to give up their electrons, so they are good reducing agents. Okay, and the terminal electron acceptor is, of course, oxygen. Oxygen oxidizes things, so it's actually a pretty great oxidizing agent. The chloroplasts, right, again, I just want to reiterate here that we're talking about the light reactions of photosynthesis. In the chloroplasts, the initial electron donor is water, right? And water does not want to be oxidized, so it's a poor reducing agent. And because it doesn't want to be reduced, it doesn't want to be oxidized. Since it doesn't want to be oxidized, it doesn't want to reduce anything. It doesn't want to be oxidized to oxygen. It requires energy, and that energy comes from light. The terminal electron acceptor is NADP+, which of course turns into NADPH. Now, we mentioned earlier that both of them establish proton gradients. But how is the proton gradient established in each one? Well, in the mitochondria, there are two things that contribute to establishing the proton gradient, whereas in the chloroplasts there are 3 different things. So in the electron transport chain of the mitochondria, the one thing that contributes to the proton gradient is the pumping of protons from the matrix to the intermembrane space, and this is done by complexes 1, 3, and 4. So if they're taking protons from the matrix to the intermembrane space, what's happening? Well, we're taking protons out of the matrix, so we're lowering the proton concentration in the matrix, and we're taking them to the intermembrane space, so we're increasing the H+ concentration in the intermembrane space. In addition, the other thing that establishes the proton gradient in the mitochondria is making water, and that occurs in the matrix. So if we're forming water by reducing oxygen, then oxygen needs to pick up protons. That's happening in the matrix. So what this does is this lowers the proton concentration in the matrix because the oxygen picks up these protons and then forms water. So those protons are no longer free floating in the matrix. So what's the net result here? We have a low concentration of H pluses in the mitochondrial matrix, which would be a high pH. And then we have a high H plus concentration of buildup of protons in the intermembrane space, which would mean a low pH. Okay, so that's the net result overall from these 2 processes. Now, in the chloroplasts, we also have a proton gradient established, but it's established in a different way. The first thing that happens is the splitting of water in the thylakoid lumen. So if we take water, we oxidize it, right? We break it up into oxygen and protons. And those protons are free floating, and if this is happening in the thylakoid lumen, then these protons would be, the proton concentration in the lumen would increase. So we'd have a higher concentration of protons in the lumen. The second thing that contributes to the gradient is the electron flow from photosystem 2 to photosystem 1. So this is when plastoquinone pumps protons from the stroma into the thylakoid lumen. So they're taking it out of the stroma, so that's lowering the concentration of protons in the stroma, and taking them to the thylakoid lumen, which would increase the proton concentration in the lumen. The last thing is that NADP+, right, is reduced to NADPH, right? And this happens in the stroma. So if these protons are in the stroma, they get tacked onto NADP+, that means we're taking protons out of the stroma, essentially. So we're lowering the H+ concentration in the stroma. So what's the net result? Is that we have a low concentration of H+ in the stroma, which is, of course is a high pH, and then we have a high proton concentration, or a low pH, in the thylakoid lumen. Last thing I want to mention is what are the differences in the energetics of each process. So in the electron transport chain, every single step of that transport chain is exergonic. So since every step is exergonic, then the overall process itself, I was going to write overall. The overall process itself is exergonic or spontaneous. And the phosphorylation of ADP to make ATP is basically just, it's powered by the establishing of the proton gradient, it's powered by the proton gradient, or the protons flowing back down the gradient. But how is the gradient established? Through these redox reactions, these exergonic redox reactions. So this is called oxidative phosphorylation, right? Because these redox reactions powered the establishing of the gradient, and all of them were exergonic steps. In the chloroplast, however, in the light reaction, there are 2 steps of the light reaction themselves that are endergonic, sorry. 2 steps of the light reaction are endergonic. There are 2 steps, right, when we shine the light on the reaction centers, that in order to oxidize the reaction centers at the chlorophylls, those require light energy and are therefore endergonic. The rest of the reactions, however, are exergonic because they all have electron flow based on reduction potentials. No energy is invested. So now in chloroplasts, we have some reactions that are endergonic, 2 of them to be specific, whereas the rest are exergonic. Now, but what is the overall process? Well, if you sum up the delta G's from all these reactions and all these reactions, what you end up getting is an overall endergonic process. The entire process itself requires light. And so because of that, essentially what they call it here is the phosphorylation is driven by the energy of light and is therefore called photophosphorylation. So I hope that video kind of sums up the sort of general idea as far as these similarities and differences go. Thanks for watching.